let's begin to try to stay a bit on schedule. So it's my privilege to introduce the first speaker. Dr. Peter Shizgal is a behavioral neurobiologist who studies brain mechanisms of reward, motivation, learning, and decision making. His research combines behavioral, electrophysiological, neurochemical measurements with mathematical modeling and optogenetic perturbation of neural signaling. Shizgal is a full professor at Concordia University, holds a Concordia University Senior Research Chair. He's a graduate of McGill and, of course, the University of Pennsylvania. He spent sabbatical leaves at the Department of Neurobiology and Behavior at Stony Brook University and at A. Martinez Center for Behavioral Imaging at Harvard and MIT. This is my dyslexia coming out. Peter's a fellow of the American Association for the Advancement of Science, the Association for Psychological Science, and the American Psychological Association. He's a recipient of the NutraSweet Prize, because he's such a nice guy, for research in feeding and nutrition, as well as the Pix Adrien Pinard of the Society of Quebec for Research in Psychology, and I translated that into English. Peter also asks us to say that he is very grateful for the mentorship and friendship that he received from both Rochel and Randy. And he has a second career as Randy's occasional deckhand and radar operator. Peter.
So 1964 was an interesting year. The New York World's Fair opened in Flushing. The Beatles made their first visit to America, and Randy Gallistel published his first scientific paper. Now, if we pay close attention to the publication information, we can see that this paper appeared in January of Randy's first year in graduate school. So from this, I infer that most of the serious writing, certainly the research that's the basis of this, was done when he was an undergraduate. So I had heard stories of Randy's exploits when he was an undergraduate at Stanford. Some years later, was seeking advice. I was an undergraduate at McGill. I was seeking advice from Norman White about where I should go to graduate school. And we were running through the Penn faculty. And Norman told me a story about an event that had occurred when he was a graduate student at Pittsburgh in the early 1960s. His advisor used to organize one-day workshops on burning issues of the day. And he organized a workshop on intracranial self-stimulation. So he invited the leading lights, Elliot Wallenstein, Jim Olds, the co-discoverer of the phenomenon. Some other guy who Norman can no longer remember, maybe it was Sebastian Grossman, and he invited Tony Deutsch. Now, Tony was rather well known for his aversion to attending scientific meetings. So he arranged to fade himself out of the picture and sent in his stead an undergraduate student. Randy was very young. Now, Randy presented psychophysical work that was just beginning in Deutsch's lab. Deutsch had the insight that with an appropriately designed behavioral experiment, you could actually infer properties of the transducer neurons, the directly stimulated neurons that gave rise to the rewarding effect. And the Goliaths who were presenting at this meeting were having fun. Wallenstein apparently was gentlemanly, but very critical. Olds was scathing and contemptuous. But neither of the Goliaths took into account the relish with which Randy engages in intellectual combat, his enormous skill as a debater, and despite his tender years, the tremendous knowledge he had already accumulated. stimulated. And the consensus in the room was that David had won the day, and the Goliaths left with their tails between their legs. So what is this phenomenon about which there was such a fuss? This is a reenactment of the discovery 10 years earlier at Yale. Hebb staged this when he brought in the reporters. And what you see depicted here is the observation that gave rise to the field. Due to a botched surgery by Olds, an electrode tip ended up in the septal area. And the experimenters noted that the rat kept coming back to the location in this box where it had last received stimulation. They seized on this phenomenon, but unfortunately McGill had no operant chambers. Hebb was no great admirer of Skinner. So Olds stayed all night and built one. And when his colleagues showed up in the morning, they would have seen something like this, which was a rat pressing a lever to stimulate its own brain. I'll show you what that phenomenon looks like. You'll see a lever come out of the wall. This rat is just being trained to hold that lever down for a second in order to trigger a train of current pulses. And then when the pulses appear, you'll see them here with the stimulated action potentials appear. See, the rat is really interested in this one. Here's a mini version of what Olds and Milner observed. The rat comes back to the location where he has received the stimulation, but he learns how to trigger it, and here he's trying to rip the lever out of the wall. So, this is the obvious question that comes to mind. The stimulation is of no value to the animal. The animal is not in a neat state. Why is the animal so interested in triggering the stimulation? Well, Olds gave an answer of a sort in a widely cited paper that appeared in Scientific American shortly after the discovery, basically saying that the rat presses the lever because it makes him feel good. Young Gallistel was not buying this. Because, in effect, talking about what the rat feels, assuming that we could measure that, is not an explanation of why the animal presses the lever. Maybe this had something to do with the sharpness of Olds' comments to Randy. And he says, to say that the sensations produced by electrical stimulation are pleasurable does not explain how these sensations operate to produce learning behavior. And the ideas of Deutsch that Randy was advocating at the time were really mechanistic ideas. They were ideas that you could simulate. In fact, Deutsch actually built a mechanical embodiment of the theory that exposed to the inputs that the rat is exposed to did the appropriate things. So the fundamental question that Randy raises in this paper is how the electrical shock acts on the central nervous system to organize the animal's behavior in such a way that it can reproduce the actions which led to the shock, which is a specific way of asking what is the physical basis of reinforcement and instrumental learning. Now, I didn't meet Randy until some years later, until I came to Penn. Randy, by that time, had been a professor at Penn for 5 years and had established a vigorous research program. And when I met Randy, he was probably doing something like this. Here he's training a rat to run down a runway in order to trigger rewarding stimulation. And I remember asking myself at the time, is this the smartest person I've ever met? I joined Randy's lab the next year, and as others have commented, it's an amazing experience to start to work with Randy and Rochel. You're suddenly accepted into this warm intellectual family. You immediately learn that they've got a place for you in their home, they've got a place for you in your heart, and that in their hearts, and that is forever. There were discussions that were very deep about science and about much else, really no boundaries. And these occurred in many places. They could occur in the lab. Here you see one of these pauses to which Jim referred. Randy is receiving information from the home planet. This is 15 years later. Here is brother Jim. And we're reminiscing about this golden period at Penn, which was golden not by accident. It was golden because of the way the faculty invested in us, invited us into their homes, the effort they put into their teaching. Paul, I'm looking at you. It also was golden because of the kind of people who are our fellow students. Jim, Susan, Mary, really it was an amazing group. This is 15 years after that. And this is just a year and a half ago. The people with us in this image are two of Randy's intellectual grandchildren. This is, excuse me. This is Ivan Trujillo Brisanti and Marie-Pierre Cosset, who are both PhD students in my lab, and it's their data that I'll be talking about today. So I'm now going to turn to the dopamine neurons and say a few things about Randy's role in demonstrating their importance in the world. This is a side view of a rat brain. And immediately we can see one problem with the pleasure center idea of olds. The sites shown in these dashed ovals where the stimulation produces rewarding effects are distributed throughout the brain, more or less from bow to stern. This is the medial forebrain bundle, which is a heterogeneous collection of fibers that interconnects forebrain, midbrain, and hindbrain structures. The electrode that produced the effects you saw in the video is here. And at the tail end of this bundle in the ventral tegmental area, we find, among others, the cell bodies of dopamine neurons, which are visualized here using a fluorescent antibody to the rate-limiting enzyme of dopamine synthesis. Now, the VTA dopamine neurons here project to ventral and dorsal ventral striatum, to basal forebrain, and to the cortex. There aren't a lot of them. If we take all of the midbrain dopamine neurons in a rat, you've got maybe 20 or 25,000 on each side. In a young human, you might have 200 to 300,000. In a human my age, you might have about half that. But their small numbers are in sharp contrast to the large number of psychological functions that are attributed to them. That's an understatement. So this particular claim is one for which Randy did the best of the early work. So in this paper, he and Don Edmonds, a fellow student at the time, developed a really good method for asking a particular neurotransmitter played a role in the rewarding effect. And Randy and his group pursued this idea in a really memorable way over the following years. The approach that Deutsch pioneered and that Randy did so much to develop and inspire was really a psychophysical approach, where we designed behavioral experiments that would allow us to infer properties of the neurons we were looking for. And that approach was adopted by me, it was adopted by John Yeomans, and we developed this entire portrait of these neurons. And that put us in the position of asking a kind of Groucho Marx question of our colleagues. Because our colleagues knew something to be true which we knew not to be so. But it seemed perfectly obvious. You see, the problem, you can see in the side view of the rat brain, is that the dopamine axons project right past the electrode. And Randy's experiments and others showed that dopaminergic neurotransmission was necessary in order to get the phenomenon. And in fact, when you manipulated the transmission, the expected things happened. So if you boosted dopaminergic transmission and you wanted to keep behavior at a certain level, you could make the stimulation weaker. if you blunted dopaminergic neurotransmission and wanted to maintain behavior, you had to strengthen the stimulation. All this made sense. So it's obvious, it's the dopamine neurons, right? But there's a problem. They have really skinny, unmyelinated axons that are very hard to excite with extracellular stimulation. In fact, it was well known from electrophysiology that the levels of stimulation we used in our experiments were insufficient to activate these axons. And it later became clear that there are many, many pathways that pass by the electrode tip, some of which are highly excitable, which are much better likely substrates for the effect, and that are interconnected with the dopamine neurons. So the one thing I want you to take away from this maze is that, in fact, the principal activation of the dopamine neurons is indirect, it's transsynaptic. Okay. In these early years, Randy was really laying out a way of thinking about how you analyze mind-brain behavior that really corresponds to what David Marr very memorably wrote down about a decade later. And the idea is this. Let's imagine that you're a reverse engineer. Okay? So reverse engineers have the job of taking a product produced by a competitor, figuring out how it works, and then presumably gaining some market advantage from this knowledge. Now, if you're given this black box, and you have to find out what are the principles of operation of this black box, what's the first question you ask? Well, you ask, well, what's it for? What does it do? Why does it do that? And then, once you've asked that question, you have to ask a rather abstract question about what the principles of operation are. We're talking about a brain. We want to know how this brain represents the external world. What information does it draw? How does it transform this information? How does that information get into memory? What computations are performed on it? And then finally, we go to the physical level. And we realize that there are many different possible mappings between the algorithmic level and the physical level. You can implement a certain algorithm in many, many different ways. And this made Randy really stand out from his contemporaries who were very much focused on the physical level. And this was at a time when many people would look at you funny if you said that rats actually had representations of the world inside their heads. And even people who would admit that maybe rats had them felt that they were irrelevant to the problem of figuring out at the physical level what the basis of reinforcement is. But Randy's view, which was very much adopted by the people he inspired, was that you had to know what you were looking for in order to be able to find it. And if you didn't have a good idea at the computational and the algorithm level, you would just be totally lost in the maze of the billions of neurons. So this is a schema from one of my papers. But the point I want you to take away from this, what I'm doing here is building a mathematical model that draws on earlier work of Randy's and work of mine, that takes the input, this train of current pulses, translates it quantitatively into what the rat does in ways that we can test and use experimentally. But look at what's at the core of the model. There's a representation here. There are aspects of the world that the animal has coded and has written down and committed to memory. So what I mean by reward intensity is the animal's subjective appreciation of something like the concentration of a sucrose solution. The animal is recording what is the probability that he'll get paid when he does what I've trained him to do. He's recording the exertion that's required in order to secure the reward, the time, extraordinarily important in Randy's formulations. And these quantities don't just sit there, they enter into real computations, and it matters what these computations are in order to derive the internal variables that are going to be used to control the animal's decision. What is the payoff I get from what the experimenter is offering me? What's the payoff from anything else that I can do in this environment? Now, Randy rather deeply probed these internal represented signals in a way that drew him into the literature on learning. And it was that voyage that led to, if I'm not mistaken, his fascination with timing, and eventually his development of this information theoretic view, this tremendously powerful and original view that he has very eloquently with his collaborators communicated in a series of very memorable books and papers. Now, what I'm going to do is to now make a big switch. And I'm going to put Randy's view in relief by talking about its antithesis. So I'm going to present an alternative view, which is actually the dominant view among my colleagues, of how reinforcement learning works, and particularly about the dopamine neurons. And then I'm going to tell you about an experiment that poses some challenges to this view. So the idea of temporal difference reinforcement learning was developed first in Rich Sutton's PhD thesis. And it comes from a machine learning tradition. Rich is a computer scientist. He had been working with Bellman's dynamic programming, and he had this insight that he could develop an algorithm that would approximate the optimization that dynamic programming gives you in a much less resource-intensive way. And the whole aesthetic here is extraordinarily spare. These guys, they wield Occam's razor like a chainsaw. Randy's view is so much closer to the biological world, which is a world with tremendous variation, where in fact variation is the raw material from which evolution selects, right? And in Randy's world, you have a rich set of primitives from which you build brain mechanisms. This is a whole different view. This is a view that things have to be mathematically clean and as simple as possible. Okay, so I'm going to illustrate this, but I'm going to illustrate this in a way that is really a caricature. It's the caricature that most of my neuroscience colleagues carry in their heads. The view that Rich and his colleagues carry in their heads is greatly more sophisticated. So what the algorithm tries to do is to predict the discounted sum of future rewards. And what drives learning is a discrepancy, a prediction error, which is a discrepancy between what you're expecting and what you actually expect, what the outcome is. And that prediction error is used to learn the value of different states of the world and the values of the actions that are available in a given state. Now, the computer scientists are not afraid of having lots and lots of states. Pattern recognition, they're very confident they can do. So they're going to put a lot of weight on the states. They're going to try to stay away from representing relationships between the states as long as they can. Now, in neuroscience, this gets immediately boiled down to predicting the reward on a single trial. And the example that's usually taught is a delay conditioning example. So the animal's in an environment where once in a while a conditioned stimulus comes on, and the timing of the conditioned stimulus is not predictable. There's a random component to the intertrial interval, that's why you use that as well. And when the CS goes off, a reward is presented to the animal. And after many trials, you start to occasionally trick the animal by leaving out the reward. So how does the theory explain this? Well, temporal differences are, if you like, a poor man's derivative. It's a difference between a signal at one moment in time showing in green and at a preceding moment in time showing in pink. And in this case, the temporal difference of the value signal is 0 because the animal has never seen this conditioned stimulus before. So at both of these moments in time, the animal doesn't have a prediction. So the difference between 0 and 0 is 0. There is no temporal difference. And then when the reward comes, it's a big surprise. If you sum the reward with the temporal difference, you get this positive signal. Here's what the animal is presumed to know after many trials. he is presumed to have learned by the temporal difference algorithm a value signal, so that the states of the world that correspond to each of these slices in time now have associated values, and those values are going to mirror the value of the reward. So, the temporal difference will now be negative at the time the reward is presented. This is subtle. When he gets the reward, what that tells the animal is there's no more reward coming for an indeterminate period of time. The intertrial interval has a random component. The instant before he got the reward, he was expecting an imminent reward. So the temporal difference signal is negative. Then he gets the reward, and that negative and positive signal sum to 0. So the temporal difference error predicts itself away. It's rather elegant in this sense, so that the algorithm finds stability. Now, when you trick the animal by not giving him the reward, you still have your negative temporal difference signal, but you don't have a reward to null it anymore, so you end up with a negative temporal difference error, which will now work to weaken the value signal on subsequent times. Okay, so how do we get from that I don't know anything about the world to I know everything stage? Well, what I showed you is that the reward prediction is just a difference signal. And that difference signal gets combined with the value of an ongoing reward. Is the person here? Okay. And the sum of the temporal difference signal and the reward is what's called the temporal difference error, which is also called the reward prediction error. This is the discrepancy. And the discrepancy does all the work. It changes the weights. Those weights are what were responsible for producing those weights are the implementation of the value signal. Now, in this depiction, which is the one you usually find in neuroscience, the weight-changing mechanism works one step back in time because it's trying to build predictions. So it's trying to change the value that you have associated with the CS one moment before. In actual fact, in computer science, they have, in the machine learning accounts, there's something called an eligibility trace, which generally has an exponential weight which declines backwards in time. But you can see how this mechanism would operate. After one trial, you have a little bit of value at the moment before the reward. So then at t-2, at the moment before that, the error signal would produce a weight change. And then the next trial at t-3, and the weight change will propagate backwards until it reaches the beginning of the CS. It can't go any farther because the jitter of the intertrial interval causes the prediction errors to cancel out. And what you end up with is this unknowable TD error at the onset of the CS. So that's the theory. But that theory doesn't make an animal do anything. The animal is just contemplative. It's sitting there making predictions about the world. So the whole idea of the machine learning researchers were they wanted to build intelligent agents that would learn from their interactions with the world and learn to optimize their future discounted sum of rewards. So this is one of the architectures that emerged to get the agent to actually do anything. The temporal difference mechanism is called the critic. It's a critic because it's telling you how bad your predictions are, but it's also telling you whether you've got your priorities straight in terms of your actions. So in each state of the world, there are these different actions you could take, and the prediction error is used to tune the probability that you'll take each of these actions when you're in a given state. And once you finally have learned everything there is to learn, and there's no more prediction error, then you'll have a fixed set of unchanging probabilities here, which is called your policy. Okay, that this error signal is not only driving your predictions about the world, it's controlling what you do about it. So the key features. Typically and historically, temporal difference reinforcement learning has been used as a form of model-free learning. This is the heavy use of Occam's razor. The agent recognizes all these different states, it learns their values, it learns actions available in each state. So it learns state action pairs, but it doesn't learn the relationships between states. There's no map in here. Both the world and the agent are noisy, and because of that, we're going to need lots and lots of trials to average through the noise. So learning will typically be gradual. In all of these accounts, you see the learning coming up slowly. And the crucial thing is that in the presence of reward prediction errors, there must be change. The reward prediction error changes your expectations about the world, and it changes your policy, the actions you're going to take. You cannot get the stability of prediction or action in the face of continued reward prediction. Okay, so now we jump forward, and we come back to the dopamine neurons. So Wolfram Schultz had been doing electrophysiological experiments in monkeys. This is a side view of the macaque brain. And he's recording in the ventral tegmental area and the neighboring substantia nigra. And the monkey gets to often see a visual pattern on the screen as a CS, and is in a variety of conditioning paradigms, like that delay conditioning paradigm, receiving fruit juice as a reward. And this is a figure many of you have likely seen many times. In this case, the animal gets an unexpected reward, and the dopamine neurons fire in response to the reward, to the fruit juice. This is after you have trained the animal in delay conditioning for many, 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 many trials. Now, when the CS goes on, the pattern goes on the screen, we get brrr, and the pattern stays on the screen for 2 seconds, then he gets his fruit juice, nothing happened. The reward response is null. And we get to see the supposed signature of the negative temporal difference signal by omitting the reward. And you see this little dip. The dopamine neuron becomes silent at the time of the reward. So basically, we take the temporal difference reinforcement learning algorithm, and we just map the reward prediction errors into the corresponding activity of the dopamine neurons. And that insight says this is implemented by the dopamine neurons. Now, the dopamine neurons that Schultz recorded from project to the striatum and ventral striatum. The dominant cell type there is a medium spiny neuron. Here you get to see what the people influenced by machine learning in neuroscience think is the basis of learning. So a medium spiny neuron has lots of little spines on its dendrites. And those spines get descending inputs that are mapped topographically from the cortex that go from the head of the spine. And then on the neck of the spine is a dopamine synapse, as if it has its hand on a tap, and it's controlling how well the head of the spine is talking to the dendrite. And here we have the electrophysiological data that seems to support the basic idea that the way learning occurs is by adjusting these synaptic weights. So this is the effect of repeated glutamatergic input in the presence of dopamine. We get potentiation. This postsynaptic effect is getting bigger. And this is the effect of repetition of the input. In the absence of dopamine, the postsynaptic potential gets smaller. We have depression. Okay, game over, we got it? Maybe not. I should say that the hypothesis is really a symbol. So many of my colleagues regard this not as a hypothesis, but as a fact. Let's move on. It has its own histories now in studies of the history and philosophy of science. So the people who brought together the dopamine data and the reward prediction area are Reed Montague and Peter Dayan, who at the time were postdocs in Terry Sanowski's lab. And Reed and Terry wrote a paper, their second paper on this phenomenon, which is based primarily on intracranial self-stimulation. So what did they say about that? Well, essentially they said, we've explained intracranial self-stimulation. The electrode is a dopatrode. the electrode is causing dopamine to be squirted and to produce non-nullable temporal difference errors, reward prediction errors. And I had some problems with this. As I've already told you, the rewarding effect of NFB stimulation doesn't arise from direct activation of dopamine neurons, but from indirect activation. There's lots of physiological data that we obtained over the years about the transducer cells that are incompatible with dopamine neurons. The second problem is algorithmic. And this is the deeper problem. If this error is really unconditional, can't be null, what that means is the rat presses the lever, and the dopamine neurons tell him, that was better than you thought it was going to be. Up your weights. So then he presses again. Oh, that was better. Better up your weights some more. And more. And more. Now, the equation in the original paper predicts that the weights go to infinity, which is a very bad number. But, you know, these guys are really smart. I mean, they'll choose an appropriately saturating weight changing function, and when they did, the prediction is quite straightforward. Small rewards should drive the animal to saturated weight values over a long period of time, over many repetitions, and a large reward should do it quickly. So I went to visit Peter. I was in London. I went to visit Peter Gelman, and I said, look, I've got a problem with your theory. I think it's wrong. But I think I know how we might fix it. And instead of throwing me out of his office, the gentleman that he is engaged in a really deep and long, wonderful conversation, which was the beginning of a terrific collaboration. And we agreed that all we needed really to do was to move the electrode so that it activated the primary reward signal. And now the temporal difference, the prediction, could null that signal. And at the time, the electrochemical data that were available suggested that the dopamine signal that was produced when the animal was self-stimulating went away quickly. So we thought, you know, maybe we're not so far apart. Then Joe Cheer, we did the experiments with a bit of timeout in between stimulations, and he reported that every time he stimulated, there was a dopamine transfer. That's a big problem. Because if the reward prediction errors are persistent, behavior can't be stable. And we've gone through this argument. Performance has to be driven to an asymptote. And we knew from decades of work with the phenomenon that we can produce stable performance at any level. The experiment that Joe did wasn't done over sufficient duration and in such a way that we could be absolutely sure that it was a killer. We intend to redo this experiment. But in the meantime, we've done something much more direct. And that's to go to optical stimulation of dopamine. So the problem that is the original sin of electrical stimulation as a research technique is its lack of specificity. It is a wonderful and important technique. It is a crucial technique for getting causal information, for asking if an injected signal mimics a signal. But in the very first paper about electrical stimulation of the mammalian central nervous system in 1871, Fritz and Hitzig agonized interpreting their data as to whether the movements they produced by stimulating what we now know to be the motor cortex were due to local neurons or fibers of passage. In the last 10 years, methods have been developed that allow us to optically activate neural populations of our choice. So we cause a light-sensitive protein to be expressed in a certain neural population, and then on a spike-by-spike basis, we can trigger these neurons. The protein itself is trafficked to the membrane. It is a cation channel, so it allows sodium and other small cations to pass. It excites neurons. And here you see the expression of channel rhodopsin 2, such a sensitive protein. You can see it's been trafficked to the membrane of these neurons. By doing double immunocytochemistry, we can ascertain that these are indeed dopamine neurons. And by using a combination of transgenic animals and viral transfection methods, we can restrict the channel rhodopsin to the dopamine. So Ivan and Marie have been pursuing this. Now, by putting the light here, we argue that we do have an unconditional activation of the dopamine. So what you see in the circle is corresponds in size to the optical fiber that's implanted in the animal. So we're looking at a horizontal section, like a top view. And look at all these processes. This, here, this is the cell body of the dopamine. Look at the scales. You have a cone of light that's diverging given the numerical aperture of the fiber. It's almost inevitable that we're stimulating axonal processes. So we're stimulating downstream from where the inhibition arrives in the somatodendritic region. And this is why, at least in this region, we feel that the excitation can't be nulled by incoming inhibition, because we're activating downstream from where the inhibition is. So the first thing we needed to do is to make sure we're actually stimulating the dopamine neurons. So what Nahi does is to inject the virus that carries the channel that helps into the gene. That gene is fused to the gene for a jellyfish fluorescent protein, which allows us to see where the product goes. And now she has to wait patiently, because the product actually diffuses in the nerve membrane. It takes weeks until it actually gets its way down to the terminals. And then what she's going to do is implant an optical fiber attached to a laser, and she'll put an electrochemical probe in the terminal field. And dopamine is an electroactive neurotransmitter. That means we can modify it by putting the transmitter in a potential field. We can take electrons away from it, and we can give the electrons back in redox reactions. Those electron transfers are currents, which we can measure. The oxidation current is in red, the reduction current is in blue. If you look at this in 3D, here is the potential gradient that we impose. So we go up, we come down, we produce these currents. They leave a signature, which tells us, in terms of the voltages at which we see the peak and the trough, what kind of electrochemical species we're dealing with. You can show these in false color, so that this figure is composed of 200 vertical lines, like this, with the vertical axis here color-coded, and the blobs are the signature for the neurotransmitter. If we now take a section through the oxidation blob, and we do some post-calibration, we can actually turn this into a concentration. So with this fiber, which is about 1/5 of the diameter of a human hair, so it produces very little damage, we can record in the brain the local changes in dopamine transmutation. And here are changes produced by light. They're very orderly. Here we're changing the strength of the optical stimulation by changing the pulse frequency during the 1 second train. And it gets stronger as the frequency goes up, and then weaker when we go over the top. Here we're changing the optic power, and you can see, again, how orderly the response is in an anesthetizer. Now what Ivan has been doing, he starts off the same way by injecting virus. He implants the optical fiber chronically, but he has the animal stimulate itself. This animal is working for light. He seems quite interested in what this lever does for him. You might even see a little blue around the connector as some light leaks out. Okay, so we're going to run the animal through what we call frequency sweeps. The animal has to hold the lever for a second, the lever goes away, comes back. Its time in the cage is 120 seconds per trial. What we mean by a trial is a period of time when the experimental conditions stay the same. The condition of interest here is the frequency of those optical pulses. As you saw before, as we raise the frequency, we increase the amount of dopamine. So we start off with a high frequency, and then we step down. Okay. Now, what I'm going to do, if I can regain control here, is to show you a simulation that my colleague Peter Dion did using a really impressive model that they derived from our behavioral data. And this is, if you like, Peter's model of the Shizgal conjecture. It's what happens if you can't null the dopamine release. So with a weak reward, what we're plotting here is cumulative work time. So the animal is putting time on a clock. And if the reward is very weak, he starts off not working that quickly. But of course, each train tells him that the reward was better than he was expected, so he starts working more and more until when the reward is strong, he gets up to that maximal rate. Now, Peter is imagining that there will be some way that the brain will find to turn off the dopamine release. If it did that quickly, then what you would get I need to retreat to this location. Okay. You will get this diverging set of straight trajectories. Because essentially what happens here is the reward prediction error launches the animal on a trajectory that's related to the size of the dopamine transient. But since then the brain, by some mechanism that's unknown, somehow turns off the dopamine release, the animal just continues along that trajectory. Since he has no prediction error on a negative or a positive one, he doesn't change his weights, and he just keeps working. And what do we get? That's what we get. So, either the brain has found some way to turn off the dopamine release, or the theory has a big problem on its hands. Now, we went further, and we developed a paradigm that could not only look at evaluation, it could also look at prediction and update. So, in this paradigm, the trials are arranged in triads. Now, the central trial, the stimulation can either be strong, middle, or weak. These are optical pulse frequencies. And there's no signal to the animal telling him what the condition will be on the trial. These three conditions are equally probable. The animal has to sample the reward to find out what's on offer. The bracketing trials are fixed. So in this configuration, in the lead trial, he always gets the strong stimulation. And on the trailing trial, he always gets the weak stimulation, and it recycles. The actual setup, we have one lever for the bracket trials. So it comes out, and when he presses it, a light goes out over it. And in this case, he gets a high-frequency pulse train. Then we have an intertrial interval with a random component. It has a flashing light to say, "Rat, you are in an intertrial interval." The levers are retracted. He then goes into the test trial. Second lever comes out. And in this case, he has to sample the lever to find out what the reward is on offer. Then there's an intertrial interval. Then he goes into the trailing bracket, another intertrial interval, and it recycles. You do this over and over. So I'm going to show you some data. A lot of the analyses and graphics were developed by Kent Conover. This is our version of a cumulative record. You see literally everything the rat did. So when he is holding the lever down, we have color. The cumulative work time is shown in color. So we start off in blue and we go to red. The triangles are when he gets a reward. The vertical position of the triangle is just jittered in order to be able to see the triangles. And you can see that he works pretty steadily. He takes the occasional break. And there are some trials where it takes him a while to get going. Here's a complete set from the experiment of individual trials. Here's the lead trial. You see he's working like crazy, although he did sit one out. He works very steadily for the strong reward, an intermediate way for the middle reward, and although he works a little bit at the beginning of the trial with the weak reward, he stops. And on the trailing bracket, he doesn't even really get going very much. There's very little bit. We can summarize those data over many trials by calculating the proportion of available time the animal is working, and then we can fold those together so that you can see that we have a high level of work, a middle level of work, and a low level of work. And the salient thing here is that the reward prediction error theory predicts that in this middle case, which is non-asymptotic performance, this should accelerate and go up. It never does. It sometimes sags a bit at the end, but it never accelerates. These are the cumulative figures, and you can see the straight, non-accelerating trajectory in the middle of the wheel. Now, I'm going to look at the issue of updating here. So how many trials does the rat need to experience? How many rewards does he have to see in order to update the information in his representation? Is it a slow incremental learning? This is shown over 10 rewards. It comes into the test trial. The median latency to press the lever is about 10 seconds because it could be weak, it could be strong, it could be in the middle. He sees one reward, the latency goes to 1/3 of a second, and it stays there. So there's one step update. You can also look at what the rat knows about the paradigm. If the rat's world, if his states are defined only in terms of the stimuli that the animal can perceive, the stimuli are identical on leading and trailing bracket trials. Same levers, same light, same levers. So if that were the case, his expectation coming into at the beginning of the trial should be the same. It's the same stimulus configuration. I'm afraid not. These are the cumulative distribution functions for the leading and trailing bracket trial. So each of these numbers is the proportion of latencies that is shorter than this or equal to the value on the x-axis. You can see that by 3 seconds here, we've got 80% of the distribution on the leading trial. On the trailing trial, we don't get to 80% until somewhere after 100 seconds. So this is before the animal has ever seen the reward. So clearly he knows something about what kind of trial he was on. In terms of updating on the test trial, here's what the cumulative distribution functions look like. So he comes into the trial not agnostic, he doesn't know what he's going to get, he sees one reward, bang. Wow. Okay. So it's obvious that the voltmetric measurements are going to be crucial and determining. And we're working hard on this. We've now been able to get, this is a recording from a behaving animal who's undergoing electrical self-stimulation. I got an email message as I was getting on the plane yesterday that Marie had succeeded doing this in an animal working for optical stimulation. We have trained animals with headgear. I just can't wait to see what these results are. This is with a chronic electrode, which can produce dopamine recordings for many weeks. So it should be possible, using this electrode developed by Paul Phillips, to actually measure the dopamine release as the animal learns the paradigm. And that will be a whole different set of questions. Okay, so some interpretations and parting thoughts. Peter Dye has been the most wonderful collaborator, incredibly generous, very thoughtful, and he will hold my feet to the fire because we are coming at this from 2 different points of view. So I'm going to give you a little bit of the sorts of ideas that the person who probably has thought as much or more as anyone alive about the hypothesis that dopamine implements reward prediction errors, what he would say. So the stable non-asymptotic performance while we repeatedly stimulating dopamine neurons, that's a big problem. Very rapid updating, that's a big problem. Acquisition about knowledge of the trial structure may be good. Let's look at the RL framework. The first thing that the machine learner would go to is to say, let's be a little bit more flexible in our Markov decision process. What if the state, we give the state a few bits to record historical information? So the idea is that if you were in this intertrial interval for a trailing bracket trial, and you could somehow remember that on the previous trial, the test lever was out, and then you could work your way back to the trial before that and know that the bracket lever was out, you would then be in a unique state, and you could associate the action appropriate for a trailing bracket trial. And if you use that similar history when you were in this intertrial interval, and knew that the trailing lever the bracket lever had been out and previously the test lever, then you would take an action appropriate to the leading trial. So they're not so much bothered. They're willing to make their view. What about the rapid updating and what to do about the dopamine transients persisting? Those remain to be serious problems. Now, you could say, well, modern reinforcement learning incorporates model-based and model-free learning. So the reinforcement learners have been aware for a long time that there are great advantages to having models of the brain. They are just very conservative in what they're willing to have the agent model. And they would claim that this paradigm is insufficient to demonstrate a true model because for them, a true model is something that supports forward search. So this is what they mean. This is a drawing by Peter's daughter, Alyssa, of an experiment done by Tolman and Honzig in 1930. So we're contrasting model-free and model-based reinforcement learning. We have a rodent in a state, a choice point, and there are three routes to a reward, a cheap one, a moderately priced one, and an expensive one. And in the model-free case, he has no representation of the relationship between the states. He just has three numbers: low price, middle price, high price. He chooses the lowest. In the model-based case, he has a map of the world. He takes the shortest route. Now we put a block in his path. So we take one of the states out of the choice set. When he goes back into the choice state, he only has two options left. He takes the one with the lowest cost in the model-based, the model-free case. In the model-based case, he takes the next shortest path. The key condition in the Tolman and Honzig experiment was to put the boulder here. So now when the rodent goes back to the choice point, in the model-free case, he has never experienced the blockade taking the middle path. Therefore, he takes it because it has the lowest cost. It's only when he can do forward search and explore the representation in his mind that he can see that there's only one path available that goes to the reward. I would have to do something like this to satisfy the reinforcement learners that we have a true model. However, we are videotaping the rats, and if they show up at the right lever at the right time. Okay, so if we win to model-based reinforcement learning, the great advantage is that you can update in one shot because of your doing this forward search. But you still have a big problem if the dopamine transients persist. So here's where Peter would go. He first says, Peter, that is me, are you stimulating all the dopamine neurons? And I have to say, no. In fact, even in the area of the infection, we only managed to get the channel rhodopsin into maybe 70-75% of the neurons. And the virus only spreads so far. I'm working with a chemist on using upconverting nanoparticles to stimulate the channel rhodopsin, and thereby using long wavelength light that can go all the way through the brain. So I think I can eventually activate a great proportion of this population. And the reason I would want to do this is because my colleague's argument is limited by a fluorophore. Dopamine neurons fire very, very slowly. They only fire at about 5 hertz. If I can stimulate half of them and I add 40 spikes per second, well, they can only go to 0. So the unstimulated population can't compensate for that. So the argument exists, but it could be countered. Also, we can see whether there is an inhibition in the dopamine neurons that aren't stimulated. We can do that with our existing methods. Okay, this is some theoretical musings and all. So when I talk to Peter, these are said to be the comments of Shizgal's evil twin. First question, could the induced dopamine burst actually function as a reward rather than as an error? In other words, does it walk like a reward prediction error, quack like a reward prediction error, but it in fact isn't acting as a reward prediction error? So the comeback would be, but then why is the response null when the animal gets a predicted natural reward? And I just point out that the information conveyed at the crucial times in the delay conditioning experiment can be looked at in an interesting way using Gallistel and Boltzmann's view of informativeness. CS onset tells the animal that a reward is imminent. These CSs are very short, they're 2 seconds in most of these experiments. CS offset tells the animal there'll be no reward for a long period of time. The dopamine neurons are somehow in the business of engaging reward-seeking behavior. Perhaps this informativeness would be highly true. Okay, so I'll just finish with an anecdote. When we were hiking on Santa Catalina Island, off to the side of the trail was some random chair. We have no idea how it got. So we encouraged Randy to ascend from and to look out over his realm. And I'm asking myself, when Randy looks over the arc of a 50-year-plus career, he looks back, and he looks forward over a similar scope, what does he see? Well, this question, well, we still haven't answered it. But if we compare the sophistication of the ideas at that time to the sophistication of an idea like Randy's definition of contingency as a normalized difference between two entropies, I hope Randy is feeling a very deep satisfaction about how far we've moved, about how far the opposition has moved, and the sophistication of their ideas has developed, and about how much he has contributed to that debate. And when we look forward, if we think about the computational tools that are available to us in work and can't work, and the neurobiological tools that have become available in very recent years. I've just lifted the corner of the veil and talked about one. There's really a whole array of these extremely well-targeted tools in space, time, and neural phenotype. I think we would have to conclude that the future is looking actually quite rosy. And I think we can hear Randy's voice essentially imploring his colleagues, rethink your tired assumptions, think big ideas, think visionary ideas, and think hard about the algorithmic level. Don't just focus on the physical realization. Think about the computation and computational algorithmic levels. And I think that we're going to hear from this fellow again, and indeed I'm sure.
Peter, that was marvelous, as usual. Let's have a few questions. Since we lack a microphone, just shout out.
Okay. This is a very naive question coming from someone who's not computationally sophisticated. But some of the things that you mentioned at the end, which I was sort of thinking about as you were talking, specifically the relation to Pullman's models, suggest that there are structures that you have to look outside your domain to try to understand why there's this rapid updating, yes, and what structures may be needed actually to model or represent the situation the rat is in, independently of the stimulation that he's getting.
So here, Morris, we get into another cultural variance. Yeah. The reinforcement learning community, immediately as you start phrasing your question, would say, frontal cortex.
No.
But Dave Renish says, the cortex is a giant situation recognition device. That's sort of the way they think. But remember Randy's argument that the primitives for building these representations are found in very simple nervous systems. So the question of what the minimum requirement for a model is actually a deep question, and it may not require multi-billion neuron structures for its solution.
So just to follow up on your point, which I didn't know about, So organisms that are primitive, that we don't think can represent or model the world quite in that way, they would show the same functions that you indicated? I mean,
can you say something about your most recent bee paper?
I'm sorry?
Can you say something about your most recent paper about navigation in bees?
Well, yeah, sure. Well, I mean, first of all, that was a totally gorgeous talk.
Yes.
That material cannot be presented more clearly than you presented it. I'm in awe. And of course, for me, it was hugely stimulating. So I'm in danger of rattling on for a very long time. But one point.
I won't let you.
Yeah. So I think you know, so you cited the Tolman and Hunziker experiment, but of course your talk began very appropriately with Tony Deutsch. And as I think you know, he did gorgeous versions of that experiment.
No, I don't know, actually.
Oh, wow. You've got to go. You've got to
That's
read
in the
his.
structural basis of behavior in the book?
Yes. Oh, absolutely. He did gorgeous maze versions of exactly that logic, and much better controlled and more elegant than the Tolman and Hunziker. So you've got to go back and look at them. And they showed very clearly that the rat had this map of its environment, as you so cleverly explained it. If you put it, what he did next depended on where you put the block. And it was one trial, the rat figuring out that, well, given that the block is here, my only option is that, but if the block is here, I could go this way. You've got to go back and look at those experiments. Then so why is there this enormous resistance to model-based learning in not only in the neuroscience community in general? Well,
Let me interrupt this very briefly there. Model-based learning is part of reinforcement learning.
Yeah.
The thing that scares them is as the tree of possibility grows, the computational task grows exponentially.
yes,
And it blows up.
yes.
So they want to avoid things like that.
Yeah, of course, that's exactly right. But of course, if they're willing to stipulate an actual complete map, then the computational problem gets much simpler. So why are they so reluctant to stipulate a complete map? Well, that's because there's no story about, a complete map is going to involve all kinds of vectors, right? And storing numbers in the nervous system on a massive scale. And then we're face to face with the fact that we have no story about how you can store vectors in the nervous system. And that's why I'm so pumped up about Jerry Heslow's, who's sitting right here and who will be describing tomorrow afternoon, the work from their lab, because I think it points us in the direction of, it doesn't give us an answer, but it says we've been looking for memories, which is where the vectors have to live, in the wrong place. They're not in the synapses, they're in the molecular structures inside the neurons. And just to return now to your final point, you're absolutely right. We have made huge progress. It's frustrating in that since I was an undergraduate at Stanford, I wanted to know what the physical basis of memory was, and we still don't know. And I'm not sure, I mean, I hope I make it into my 90s as my mother did, but I'm not sure we're going to know before I leave the scene, and I find that deeply frustrating. But on the other hand...
You won't once you will.
On the other hand, there's no denying that we've made huge progress. We've come a long, long way, and that's deeply gratifying. So that's where we are at this time.
What about the bees?
I'm sorry?
The original question was
Well,
about the bees.
Well, it was the bees. The paper just shows that the bees have a cognitive map of their environment, and the point is, it's not the first paper to show that. but again, there's this enormous resistance within the neuroscience community to granting that fact. There's been huge behavioral evidence in favor of this now for decades. And the vertebrate level, because of the electrophysiological evidence on the hippocampal cells, once you look at a boundary vector cell or a grid cell, it's game over. Okay, the animal has a map of its environment because the firing of those cells is clearly being driven by the learned geometry of the environment. And that's now, Ken Chang's talk will speak to exactly this. So among vertebrates, or at least rats, everyone agrees about that. But so the idea that the insect brain, right, which has fewer neurons in the total brain than you have in the hippocampus of the rat by a couple of orders of magnitude, the idea that that brain with less than 1 million neurons also has a complete map of a 5-kilometer square environment. Whoa, that's a really hard sell. And what this B data is showing increasingly, and again, it shows progress because co-authors on that, the manuscript came from the Menzel lab. 20 years ago, I was arguing on the basis of the bee navigation data that the bees and the ants had a map of a large environment. And at that time, all the guys who were actually doing the experiments, including Menzel, didn't agree with this. And Menzel and I had a long, all afternoon lunch in which we argued very cordially back and forth about it. Well, some people sometimes change their minds, right? Jerry changed his mind because of his own data, and eventually Randolph Menzel did too, right? And just like Jerry, as soon as he went over to the dark side, then oh my God, my own data require me to assume these bees have a map. Then, like Jerry, he couldn't get published, right? Because all of his fellow guys, he was an apostate, right? And they dump on him every chance they get.
I think there's something deeper.
So you wanted to say something?
Well, there's just a sociological fact, which is fundamentally the scientists involved here and in human cognition are still dedicated to an associationist account. And in that case, what are you talking about? A model in the head that gets used to plan?
Because weights don't make a model, right?
Right? It's used to plan and make actions, and if the actions are erroneous, well, then I didn't do what I thought I was going to do. I'll try again on the basis of the model. So there are constraints on what the options are going to be. And, you might not get it right the first time, but you're not going to take a whole lot of trials.
We're partway there.
Yeah, but that seems to be.
We're partway there. The idea that model-based learning is now accepted widely in the machine learning community.
Yeah, right.
Among neuroscientists who do reinforcement learning. But the primitives for building the model are still the same old ones.
Yeah, right.
Randy's point of view is you can't build those models out of those
Exactly.
primitives.
Exactly.
Yeah, but it is a sociological fact, I think, that people are committed to this, particularly in the U.S., England, and Canada, for political reasons. I mean, it's a foundational assumption about the nature of who we are. Everybody's equal, you learn by experience, and so on. Frank states.
Why don't we take one more question since this is such a rich conversation? And then why don't we take a 5-minute break like we did yesterday because it seems to please the audience. One more question? In the back.
Yeah, Peter, you mentioned that the light might not be stimulating all the dopamine neurons, but in a natural learning situation, what would you predict would be the percentage of dopamine neurons activated?
I don't think we know the answer to that because you have to know which ones were silent and didn't respond. It would be interesting to ask Wolf on that question. I guess there's enough of this. The dopamine neurons have 3 states. There's off, there's steady, slow firing at around 5 hertz, and then there's this bursting state. Now, the ones that are off, how do you measure them? So you have to do something with drugs to wake them up to know that they're there, and then count them in some fashion. Tony Grace used to do it by counting how many you got in the electrode track. I need to think about it, Mark, a bit to see how you would actually do that. But I should mention that there are some very interesting molecular techniques now where you give the animal a natural stimulus. That activates an immediate early gene product. And you use the immediate early gene product to target the light-sensitive, optically activated protein. That may be a way of getting at the more naturalistic coding that you're asking about. Because this driving everybody together is artificial, and you're right that it may be a problem.
Let's thank Peter again. 20 up now, if we could be back by quarter of 5 minutes. Coffee, bathroom.
There are structures that you have to look outside your domain to try to understand why there's this rapid updating. Yes. And what structures may be needed actually to model or represent the situation the rat is in, independently of the stimulation that he's getting.
So here, Morris, we get into another cultural variance. The reinforcement learning community, immediately as you start phrasing your question, would say, frontal cortex.
No.
But Dave Reddish says, the cortex is a giant situation recognition device. That's sort of the way they think. But remember Randy's argument that the primitives for building these representations are found in very simple nervous systems. So the question of what the minimum requirement for a model is actually a deep question, and it may not require multi-billion neuron instructions for its solution.
So just to follow up on your point, which I didn't know about. So organisms that are primitive, that we don't think can represent or model the world quite in that way, they would show the same functions that you indicated?
I mean, can you say something about your most recent bee paper?
I'm sorry?
Can you say something about your most recent paper about navigation in bees?
Well, yeah, sure.
3 states. There's off, there's steady, slow firing at around 5 hertz, and then there's this bursting state. Now, the ones that are off, how do you measure them? So you have to do something with drugs to wake them up to know that they're there, and then count them in some fashion. Tony Grace used to do it by counting how many you got in an electrode track. I need to think about it, Mark, a bit to see how you would actually do that. But I should mention that there are some very interesting molecular techniques now where you give the animal a natural stimulus. That activates an immediate early gene product. And you use the immediate early gene product to target the light-sensitive, optically activated protein. That may be a way of getting at the more naturalistic coding that you're asking about. Because this driving everybody together is artificial, and you're right that it may be a problem.
Let's thank Peter again.